Extraordinary times call for extraordinary measures is a good way to describe the current rollout of the global COVID-19 vaccination campaign, an unprecedented medical ambition in human history. Both the speed at which it was developed and its role as the last lifeline, as proclaimed by politicians and the public, leave some people skeptical about its safety and efficacy.
In today's UTX episode, we bring you the facts and give you a clearer picture of the benefits and challenges of the mRNA vaccine in its combat against COVID, but also potential future use cases that could go much further. Let's explore. Welcome to the UTX podcast, your source of tomorrow's technology, presented today by Techniax. I'm Ani.
And I'm Valentin, and today we're going to talk about vaccines.
Hundreds of vaccines of various vaccine types are currently being developed to combat COVID-19. Around 50 million doses have already been given in over 50 countries.
We will cover what vaccines are, how they work, what other types of vaccines are being used, such as mRNA and viral vector vaccines, and how they can help us to create a utopian future. Let's explore.
So let's start by what vaccines in general are and how they work. A vaccine is a biological preparation that provides active acquired immunity to a particular infectious disease. A vaccine typically contains a biological preparation from a disease-causing microorganism or a synthetically constructed preparation that resembles it. Now, this preparation is often made from a weakened or killed form of the microbe, its toxin, or one of the surface proteins. This agent then stimulates the body's immune system to recognize the agent as a threat and starts producing antibodies against it.
In our combat against infectious diseases, vaccination is our best weapon. Widespread immunity can be achieved and has been achieved for several worldwide diseases such as smallpox, polio, measles, and tetanus. They have been completely eradicated using vaccines. The challenge using vaccines is that you want to reduce the risk of illness, but still retain the ability to induce a beneficial immune response by using the virus inside our body.
Now, let us focus in on mRNA vaccines because the current combat against the COVID-19 pandemic has brought up this new mRNA vaccine. And to understand this mRNA vaccine, we must understand what RNA and mRNA really means. So RNA, or ribonucleic acid, is a polymeric molecule that is essential in many biological roles in our body. This includes coding and decoding our genes, as well as regulation and expression of these genes. So many cellular organisms use messenger RNA, or mRNA, to convey or communicate genetic information that directs synthesis or creation of specific proteins. Now, not only organisms, but many viruses, in fact, encode their genetic information using an RNA genome.
So when we talk about RNA vaccines, our goal, as opposed to the traditional inactivated vaccines, is to not have a weakened version of the virus inside our body, but just have the protein inside it. And the protein is actually produced by the body itself. And it does that by receiving the RNA information by a messenger. And this is why we call it the mRNA information. So once inside the mRNA is inside our body, it helps the cells to build that foreign protein that would resemble the pathogen or virus. This is called a spike protein, and the COVID virus has several proteins on top of its surface, but the current one that is being used is the spike protein. It serves as the starting point for researchers to arrive at a vaccine much faster, and they can build on top of previous efforts to do that. When the SARS and the MERS outbreaks happened in 2003 and 2012, scientists already worked on that. However, these outbreaks were much shorter, so their efforts did not come ultimately to fruition. So the protein molecules stimulate an adaptive immune response which teaches the body to identify and destroy the corresponding pathogen when it meets that one again. The mRNA is delivered in by placing it into lipid nanoparticles or fat bubbles, which protect it before it reaches the cell.
So in a nutshell, an mRNA vaccine teaches the body to produce fake antigens itself so that this will provoke an antibody response to it. Now, the advantage of RNA vaccines over traditional protein vaccines are superior design, production speed, lower cost of production, and the induction of both cellular as well as humoral immunity. One notable disadvantage, now this is specifically about the Pfizer-BioNTech mRNA vaccine for COVID-19, is that it requires ultra-cold storage before distribution.
So while Pfizer-BioNTech and Moderna are using mRNA vaccines, the classical variation of vaccines is when we look at the inactivated vaccines. And inactivated vaccines use the virus itself, a weakened or deactivated version of it that is non-threatening, and thus produce the bodily response. The Chinese company Sinovac is currently in the final stages with their inactivated vaccine in several jurisdictions. UAE, for example, has already approved it. The effectiveness, however, has been shown to be a bit lower, with only 79 to 86%, which is not that much if you compare it to Pfizer, BioNTech, and Moderna, as we'll see in a second. So it's not as high as for other vaccines.
Another type of vaccines is a protein subunit vaccine, and these use the spike proteins as an antigen. Now, a major disadvantage of protein vaccines is that the immune system is less likely to recognize and attack the protein antigen as opposed to a full stack real version 100% virus. And this means that the body will create a weak immune response. Now, to counter that, oftentimes chemical agents called adjuvants are added, and depending on application, these can increase or prolong antibody production and help with the activation of so-called T cells. And similarly, actually, the mRNA vaccine also requires a second dose to be injected after some time so that the immune response can be boosted.
Another type of vaccine is the viral vector vaccines. They are using so-called adenoviruses, which are harmless host viruses, in order to transport the virus itself, or not the virus itself, but the virus mRNA into the body cell. In theory, a vector can be used to deliver the code of various antigens at once, which might prove very beneficial in the future. There's two examples currently that are quite prominent, which is the one that is being developed by Oxford AstraZeneca, which can be stored at regular fridge temperature, a major advantage against mRNA vaccines, or at least the one from Pfizer-BioNTech. And the effectiveness of it is a bit reduced to 62 to 90%. AstraZeneca, however, has said it will produce 700 million doses by the end of the first quarter of 2021 already. Another one, and actually the first one that was approved worldwide, is the Russian Gamaleya Sputnik 5. The Russia's version of a COVID vaccine also works based on the viral vector and has shown a reported 92% effectiveness in studies. Its advantage is that it can also be stored at regular fridge temperature, which has major advantages regarding logistics and cost savings.
Now, another type of vaccine that we want to showcase today is a so-called DNA vaccine. And as the name might suggest, it is very similar to an RNA vaccine because both DNA and RNA are nucleic acids. And so they contain DNA codes for which, or it contains DNA, basically genetic information. And this means the cell hosts can code for proteins of the pathogen. Now, the only difference between a DNA and a RNA vaccine is that the genetic code from an RNA vaccine enters the cell, but not the nucleus, whereas genetic code from a DNA vaccine enters the cell nucleus. And we are not so sure about this, but potentially, if something were to go wrong, it could impact the DNA of a cell. But again, we have so far not tested DNA vaccines, so this is a big potential, but as we know, they share all the advantages that RNA vaccines also have.
So we looked at quite a high variation of different vaccine types, and all of them share the potential to solve COVID. But as for now, the two vaccines that have been approved in the Western world are both mRNA vaccines. And they are, as mentioned, Pfizer-BioNTech and Moderna. In the United States alone, 6 million people have already received either of the two vaccines. The way they do it is that you need to have two doses, with an ideal timing of 28 days between them.
Now, the first mRNA vaccine, or the creators of the first one, is Pfizer-BioNTech. And they had the first ever authorization of an mRNA vaccine by the UK authorities back in December 2020. The FDA, or Food and Drug Administration in the US, followed for the very same vaccine just a week later, by mid-December, and they had demonstrated a 95% efficacy in phase 3 trials. Don't worry, we'll explain what these phases mean later on. So in total, combined with Moderna, Pfizer-BioNTech, they aim to supply up to 2 billion doses by 2021.
So one of the major disadvantages of Pfizer-BioNTech is that they require you to store their vaccine at minus 70 degrees Celsius. And this is quite expensive and has some major requirements regarding logistics, which are very hard to uphold, especially in emerging countries and humid climates. Now, Moderna has the advantage that you only need to store their vaccine at minus 20 degrees. So that's actually an innovation within mRNA. Moderna's efficacy was measured at 94.5%, so very similar to the 95% of Pfizer-BioNTech. Moderna has said it aims to ship 500 million doses in 2021. However, just this week, we have seen a major setback for Moderna because in California, trials have been stopped after a vaccine batch has caused severe allergic reactions for an abnormally high number of subjects. So the last word is not spoken related to this.
Now, why is this exciting? So, as opposed to the other vaccine types that we showcased, mRNA vaccines do not use the actual virus or pathogen that is involved, meaning that they are not infectious. So, using the pathogen as a basis for the vaccine, as we know, could bear the potential risk of a disease outbreak if something were to go wrong in the laboratory or the production facility. However, as mRNA vaccines only use the underlying genetic code of the virus or its mRNA, you can create an immune response in many people without endangering anybody along the supply chain of this vaccine production.
Another reason that this is very exciting is currently that we see all these mutations of the virus. So the virus is constantly changing with the goal to spread. It has not the goal to kill us, but to spread. And speed is thus a key for us to eradicate the virus before it accidentally or probabilistically builds something that is even more dangerous than its current version. And in this sense, mRNA has a much faster design and production process, as well as higher standardization and smaller error rates in production. So that's very beneficial.
And lastly, mRNA vaccines generally have a lower cost of production. However, AstraZeneca, which was a vector vaccine that, as we remember, had a harmless adenovirus, has been able to produce even cheaper solutions to reduce costs for storage and logistics, as they do not require these cold temperatures that mRNA vaccines do.
The current breakthroughs are based on decade-long research already. So mRNA vaccines have been have been developed or have at least been researched on. Previous use cases include flu, Zika, rabies, and cytomegalovirus, so CMV, which for all of these researchers have been working on mRNA vaccines, but no major breakthroughs have been achieved.
And who knows, in the future, maybe these mRNA vaccines could trigger immune responses that could potentially even learn how to target cancer cells. And it might be even thinkable that an mRNA vaccine could allow us to introduce our body to immune response to multiple pathogens in one shot, meaning that you could take one vaccine dose for several diseases.
Where this also gets very exciting is if you consider how proteins fold, and they fold from amino acids using sequences of mRNA. So the better we understand how mRNA works, the better our potential understanding of the 200 million proteins that are known to us is. And this is very interesting because proteins are the building blocks of life, and all kinds of diseases can be triggered and targeted using mRNA and then proteins. So to accurately predict and control this process has the potential to unlock new levels of research being applied in areas as diverse as drug design, crop enhancement, or plastic degradation. Scientists have tried to solve this problem actually for more than 50 years, so the problem of protein folding. And last November, Google DeepMind's algorithm has trained on 170,000 genetic sequences of proteins. And they managed to accurately predict protein folding with around 90% accuracy. And thus, they actually have unveiled an algorithm that can predict in what proteins that amino acids will fold into. So that will prove very useful in the future.
Now, mRNA vaccines, as we know, sounds very exciting, but like any other technology that we've so far covered in our episodes, it is not without its challenges. So, as expected, mRNA vaccines have their challenges too. Now, the first bigger challenge is that of trials and authorization. Now, we will take a deeper look into how medicines in general undergo trials until they can be authorized for general public use.
So what is true for COVID vaccines and is true for any medical treatment is that it needs to undergo clinical trials before being admitted to be marketed and distributed to consumers. And this in various jurisdictions. So the authorities of the respective countries judge the benefits and risks of the new treatment based on the validity and reproducibility of data on human trials before taking any decision regarding its admission. Trials do happen in several stages with increasing number of participants, and they may last for up to a decade because they are also lasting after the treatment has already been admitted to the market. They cost a total of up to billions of dollars. So the business model of pharmaceutical companies is very much planning based. Only around 10% of drugs going into the clinical trials ultimately come out of it successfully, so ultimately become approved.
So now we've talked a lot about clinical trials, and clinical trials are built up in 5 phases. And the last phase is usually conducted after a treatment has been initially approved. So let's walk through how a clinical trial and its phases look like.
There is a voluntary so-called phase 0, which is optional, and it involves human trials to check how the human body and the drug affect each other, so how the human body reacts to the drug, and also how the drug reacts to the human body.
And the next is phase 1, and here you have a small group of people, so anywhere between 20 and 80 people. They test the drug for safety as well as any potential side effects.
Phase 2 might be split in 2 subphases, phases 2A and 2B, with further participants, and they focus on efficacy and what is the ideal and required dosage in order to achieve efficacy in order for the medication to be impactful.
Now, once this has been gleaned, you come over to phase 3, where you undertake this trial on a much larger group. Now we're scaling it up to 1,000 to, in some cases, even 3,000 subjects. And you test for various factors, not just efficacy, but also maybe a comparison to existing treatments, side effects, and optimal usage.
Currently, the 2 vaccines approved in the Western world are undergoing phase 4, which lasts the longest. And this essentially means during the lifetime of the drug, testing for risks and benefits and optimal use is continued.
Yeah, so as you mentioned, the newly approved COVID vaccines from Moderna and Pfizer BioNTech are now in the phase of having trials with tens of thousands of people. These have been conducted, and these also include additional 8-week post-final human trials.
In the Western world, the lighthouse authorization entity is the Food and Drug Administration of the United States. The approval process of the FDA obviously influences other authorities, even though they undertake their own trials, or their own research and their own decision-making process. In Europe, we have the European Medicines Agency, and the emergency use of, like, what FDA has done is they haven't given a full authorization to the vaccines, but actually an emergency use authorization. And this means that the product remains unapproved. Either it remains unapproved or the use of an approved product remains unapproved. Mm-hmm. So this means that we only have a temporary approval of those vaccines. And the reason that this is possible is because a material threat materializes and has been declared by the Secretary of Homeland Security, which is COVID-19 last February.
Now, to quote, a consideration of a drug for an EUA, emergency use authorization, requires a finding that it is reasonable to believe that the drug may be effective to prevent, diagnose, or treat serious or life-threatening diseases or conditions. Now, the phrase may be effective is based on a risk-benefit analysis, and obviously also on the totality of the scientific evidence available. So it is precisely for such a situation when you have in the case of the US, just focusing on that, a epidemic, but of course COVID is a pandemic, where these drugs must be given emergency use to prevent lives, to thwart this very materialized threat.
And this is also the reason why this is not the first time that emergency authorization use has been issued. The first, not the first time, but one of the times was in 2009 during the swine flu pandemic for PCR tests and drug treatments versus the flu. During the COVID pandemic, this has also happened before, and it was used to pass test kits, as well as UA was given to the remdesivir drug for medication, and also the hydroxychloroquine, I hope this is correct, drug. However, in that case, so in the case of hydroxychloroquine, it has been revoked in June 2020, proving that sadly UA is not a 100% safety bet.
So moving on now to efficacy and adverse effects of the vaccine specifically, we know that the adverse effects of this mRNA vaccine are in many areas very similar to traditional vaccines. And these are known as reactogenicity. And these are usually triggered within people that have an autoimmune response, meaning that the body responds to its own healthy cells and tissues. And an example for this autoimmune-based disease is multiple sclerosis. So from vaccines, we know that adverse effects may include fever, or fatigue, or maybe sore arms, depending on where you inject the vaccine. Now, to counter this, the mRNA sequences in mRNA vaccines are designed to mimic the sequences produced by the host cells in the human body, to not force the body to trigger such autoimmune responses.
An advantage that mRNA vaccines have over inactivated vaccines is that they do not rely on egg proteins. So if you are allergic against egg, no problem here. You won't be allergic against mRNA vaccines.
Now, we know that the efficacy of Pfizer-BioNTech and the Moderna vaccines are 94.5 or 95% respectively. And this is much higher than the previous test applications for mRNA vaccines. And the only explanation here is the sheer amount of resources and testing that have been invested in the creation of these vaccines.
Some of the vaccines, such as Pfizer-BioNTech's, as mentioned multiple times before, need to be stored in ultra-cold environments, which means minus 15 to minus 80 degrees. In the case of Moderna, it's minus 15 to minus 25. And you could put it inside a home freezer. With Pfizer-BioNTech, you couldn't. So that's a major challenge for logistics, because you need to keep that cold. It's a costly logistics process. But also what is interesting and important is how long you can store these. And mRNA vaccines would remain stable for 30 days at temperatures between 2 and 8 degrees. What makes for the longevity differences and the temperature requirements is what outer protection is used, the LNP, and also what mRNA secondary structure is in use.
Now, another potential challenge or issue with this mRNA vaccine is social media and the misinformation regarding the vaccine's impact on the human body. And some sources falsely claim that the vaccine would alter a person's DNA. Again, we've said that this is an RNA vaccine, meaning that the genetic information is transported via an mRNA. This mRNA is not an actual virus. It is merely teaching the body how to produce the spike proteins, or the little knobs on top of a virus, so that the body can produce these fake antigens and then create an immune response against it. A DNA vaccine would enter the cell nucleus, and if something were to go wrong, it could impact a person's DNA. But an RNA vaccine does not even enter the nucleus, so there's no chance of it altering a person's DNA.
What would help massively against misinformation is having evidence for long-term safety, which is obviously quite hard to achieve. And it is some kind of chicken and egg problem, because if people do not want to get vaccinated, you cannot prove long-term safety. And yet many people want to wait to be vaccinated until long-term safety is proven. So that's a chicken and egg problem right there. Now on to ethical, legal, societal, and economic considerations. The whole setup of vaccination infrastructure and organization of vaccination process for a historically large number of people at once or within just a couple months includes planning, acquisition, transportation, and logistics. For instance, the cold storage of the vaccine, if that is needed, but also procurement of vaccination material, as well as training and managing healthcare professionals, and also managing their capacities, scheduling the treatment, the vaccination with the patients. As we've seen just in recent times, there were critiques against politicians because they have not ordered enough vaccine, because it was essentially a bet on which firm would first come out with a successful vaccine. And all of this is going on right now currently. And many politicians say that mistakes and procedural imperfections are to be expected because of the unprecedented nature of this adventure.
Another more societal consideration is this conflict between the freedom of individuals to choose if and when they will be vaccinated, versus the economic and social benefits of having a society that has herd immunity. Some people say 60%, but of course, the higher the number of people who have immunity, the better for the society. So there's a freedom versus security debate here as well.
Yeah, and what is underlying this is that you need to not just prove that the vaccine helps the one vaccinated, but also the others, so that it also prevents the virus from spreading further. And two questions really stand out here. The first one is, who are the target groups of being vaccinated first? And obviously, risk groups and elderly are the ones that are supposed to be vaccinated first. But then the question is also, where do you make the distinction? What is risk group? What is not risk group? And we also come towards global inequality in a second. But when we talk about discrimination, the second point is, and in this week, actually, there has been some major news on this. What happens to those not being vaccinated? Do they share the same rights than those being vaccinated? And if you ask the governments of Greece and Portugal, it's not the case, because they have just placed their claims inside the EU that for traveling, they want non-vaccinated people to basically be disallowed to travel. You can see that they have some economic interest in this, because they want to open their borders for tourists, for mass tourism as soon as possible again. But that's a major concern, and not just when it comes to traveling, but also when it comes to use of both public and private infrastructure. So currently, to our knowledge, private owners of infrastructure, they could actually enforce such a rule so that non-vaccinated people couldn't enter. However, there are also debates within politics that they would issue new laws that they could prevent that from happening.
Yeah, well, we already know about Qantas, an airline that has said that they would only welcome guests, travelers, if they have if they are vaccinated. So this is definitely a source of discrimination against people who do not wish to or who have not been able to um, get this vaccine. But we delve into the broader topic of extending or widening global inequality, because as we know, the cold storage, the timely manner, the fact that you have to take 2 doses, it costs money, several vaccines. There's a wide price range among the different vaccines, and this obviously means that the socially and economically more well-off people will get access to these vaccines earlier than people in developing countries. And this could widen the inequality gap.
It has already been claimed by some institutions like the United Nations that there is a major problem here, that the distribution of the vaccines to emerging countries is not really undertaken fairly, or the distribution in total is not undertaken fairly. So ethical considerations are coming into play as well. As we've already talked about, the idea that you need to order more vaccination, more vaccines, more doses than you can that you could use at the end of the day, because you need to assign a probability to which manufacturer is first, and you want to solve the problem as soon as possible. The nature of the time has been to handle large budgets, and one of those large budget items is to buy vaccines. And it has been a major concern in many countries whether or not to spend more or less on this. So what is the price tag that you assign to herd immunity?
That is a very good question. I think that is one that can't be answered. It can only be debated upon. We would like to mention that at the time of this recording, it is Friday, the 22nd January. And we know that, we've always known that technological progress speeds up, but this space is moving extremely fast. I mean, to just 2 months ago, things had not happened yet, and now we are very sure that within 1 to 2 months the situation will look very different again. And I think one positive takeaway from this entire vaccine and global pandemic conundrum is just the amount of human progress that we can achieve if we put aside our differences and have a global cooperation that is enabled, and you have large pools of resources that are routed effectively. It's definitely one of the better things to come from this pandemic.
Yeah, we would wish for that also in terms of other technologies and other innovation to happen. But as for now, let's look into a bright future. And the future is our utopian day in the life. And I can tell you from waking up in the morning that it's really exciting, because I'm waking up and I'm thinking about the party last night. Yes, there is parties. So I just briefly remember the times when social distancing was the only way to prevent an infectious disease from spreading. I just smile about this distant memory as if it was some medieval abstract concept that was from some previous times when technology was not that far, and start to go about my day.
Exactly. I also remembered back a few years ago when this other big virus came up and people thought it would be a major pandemic, but an mRNA vaccine was produced within a short few weeks, and we could so enjoy our summer festivals. And as I think about it, I pour myself a cup of coffee. And I just read the newspaper article in today's edition that there have been even more breakthroughs in the development of new vaccine strategies, some that even involve harmless adenoviruses to test multiple agents at once, which means that, of course, you can exponentially decrease development times to eradicate the last currently known and potentially newly appearing infectious diseases.
And now it's lunchtime. I'm meeting my roommate for lunch, and she tells me how AI-based research into protein folding has now cracked the last unknown protein sequencing processes. And this allows us to control for the synthesis of any proteins. Progress in mRNA research has greatly contributed to further enhance our understanding of protein folding and not just solve for protein folding with our algorithms, but also understand what these algorithms are doing. This means advancements in combating all major known diseases have been achieved, and the days we have lost fellow friends and family members to cancer, heart diseases, or Alzheimer seem long gone. Only a minor number of cases is left on Earth.
Yeah, I remember the words Parkinson's can only be found in medical students' dictionary books. Though it's no longer part of the general populace's vocabulary. And then in the evening, after work, I bring my children to a medical checkup at the doctor's, and we decided to give them a one-shot viral vector vaccine. And this one is actually quite new and has been tested, but it's new because it enables the cells to build proteins resembling most of the not yet eradicated germs that are still in existence. And the antibody development and responses have been well developed, documented research, and the error rate is practically zero. So there are no multiple trips, no crying children, no big fuss about all of this. It's just you go once, one shot, and you're all set.
And I close my day off by calling a colleague. Unfortunately, he still caught cancer, but his cancer treatment based on mRNA is going on very well, and he's progressing much faster than previous cancer treatments can. And his chance of surviving this is almost 100%.
Exactly, and the reason for his miraculous recovery was that in record time, his BCI was able to inform the local healthcare center that there was a cancer building, and it immediately um, instantly sent information about this cancer as well as the proteins required to combat it. And the very next morning, he got into his self-driving car, and he didn't expect his car to stop by the healthcare center, but he didn't matter because it's a self-driving car. He was working anyway. I think he was even watching a video, enjoying his time. But he told me that his computer diagnosed it in the evening, and the next day morning at 9 a.m. he had a personalized protein sequence that would treat it. So he's one of the next generation treatment batches where within 24 hours you can go from notification of cancer all the way to a personalized treatment that does not involve radiation, chemotherapy, or any other experimental treatment, and has no side effects. So it is a wonderful and glorious utopia.
Great future indeed. Yeah.
And that concludes our episode. This has been a very short episode indeed, but the topic of mRNAs, mRNA vaccines, we decided to induce it into our UTX Hall of Fame nonetheless, just because of the massive impact that we feel that these types of or this genetic developments will mean to future medicine, future society at large. And it's definitely a topic that is worthy of discussing anytime, but especially now with this pandemic, hopefully coming to a close very soon, is one that deserves its own episode.
Right, so this has been episode 8 of Utopian Techniax. You find us on YouTube, Spotify, Apple Podcasts, and all other podcast platforms via Anchor. In the future, we are also planning to do a blog. And yeah, stay tuned for our next episode.
And as always, stay utopian.
Your Techniax.